Welcome back to Dissect the Question. A researcher is trying to discover a drug to improve muscle contractility by modulating sarcoplasmic calcium. Which of the following best describes a drug that blocks a muscle protein which would result in the highest concentration of calcium in the sarcomere? So we really don't need the vignette for this. This is kind of a standalone question. But let's go ahead and dissect it anyway. We're looking for calcium in the sarcomere. How do we get a high concentration of calcium in the sarcomere? Well, we have to know where calcium comes from. Remember, we have this sarcomere, Z-lines with actin, thin filaments attached to the Z-lines. Those are the thin filaments, and then we have the thick filament known as myosin. And it's the cross-bridging between actin and myosin. When calcium comes in through the T-tubules and the ryanodine calcium release channel is responsible for that calcium entering the sarcomere from the T-tubules. And remember, that's initiated by an action potential, depolarization of the sarcolemma, which is the membrane surrounding the muscle. So, it turns out that we can look for structures within the sarcoplasmic reticulum that if we block it, calcium is going to increase in the sarcomere. So we're talking about what's going on in the sarcomere in here, not what's going on in the T-tubule, although we know the calcium is coming from the T-tubule in the sarcoplasmic reticulum through the ryanodine receptor. But now we have to use our logic. If we block, the question is asking about blocking, so if we block the ryanodine receptor calcium release channel, what's going to happen? Less calcium is going to get in here to the sarcomere. Sarcomere. Inside there. And we're asking, how do we get the highest concentration? So ryanodine receptor, even though we know it's associated with the sarcoplasmic reticulum, would not be a good answer. Then let's look at the others. It pops out at me that dihydropyridine receptor is actually in skeletal muscle and cardiac muscle associated with the ryanodine receptor. It's actually part of a ryanodine receptor, dihydropyridine receptor complex that controls the release of this calcium. So if ryanodine isn't the right answer, probably dihydropyridine receptor would do the same thing. If we block that, we'll have less calcium in the sarcomere. Now let's look at SERCA. What is SERCA? Well, if this is the calcium release channel here on this side of the Z-line in the T-tubules associated with the sarcoplasmic reticulum, SERCA stands for sarcoplasmic reticulum calcium pump. It's an active transport mechanism to get calcium out of the sarcomere. Calcium out of the sarcomere. And why do we want calcium to be removed from the sarcomere? That's for relaxation. By the way, we cannot have purposeful movement without both contraction and relaxation. So what is purposeful movement? Purposeful movement is the ability to move a load And we don't get purposeful movement without both contraction and relaxation phases. For example, a muscle spasm is not considered a purposeful movement. We consider that a non-purposeful movement because we only get contraction without relaxation. Therefore, it seems like that sarcoplasmic reticulum seems to be the best answer. But remember, we can't stop there because we always have to look for the possibility that there's two correct answers and we're always required to select the very best one. So there's two we haven't talked about, actomyosin ATPase and tropomyosin. Tropomyosin is one of the subunits of actin. We have two subunits of actin. One is troponin and the other is tropomyosin. And if we block tropomyosin, we can keep the muscle in a state of rigor because tropomyosin is responsible for relaxing the actin and myosin filaments, for allowing the actin and myosin filaments to go back to their original conformation so the Z lines can go back to their original length when we remove the calcium by the sarcoplasmic reticulum pump or the SERCA during relaxation. So there is a connection here, but if we block that protein, we are not going to have calcium accumulate because it doesn't control the release or the uptake of calcium. It just responds to the calcium. And what about actomyosin ATPase? Well, don't get tricked by that word. Actomyosin ATPase just means an ATPase that provides energy for myosin. So another name for actomyosin ATPase is myosin ATPase. That's involved in the cross-bridging. That's involved in the cross-bridge cycling. So again, the actomyosin ATPase is responding to the calcium, but it's not controlling how much calcium is in the sarcomere. So we're stuck with D, SERCA, the best answer and the most appropriate for if we describe a drug that blocks SERCA, calcium will accumulate in the sarcomere.